Hello, everyone, and welcome to today's cryoEM Current Practices webinar. You're joining the three NIH-funded National Cryo-Electron Microscopy Centers. My name is Christina Demani. I'm a scientist and training liaison at the National Center for cryoEM Access and Training, NCCAT. I'm joined today by staff members from all of the three centers, including two of my colleagues here at NCCAT, Eugene Chua and Ed Eng. Joining from the Stanford SLAC cryoEM Center, S2C2, is Nathan Burrows. And from the Pacific Northwest cryoEM Center, PNCC, is Vamsi Rayaproilu. Before we give the floor to our speaker, we'll have a quick update from each center on the resources that we make available at no cost to the research community. Today's speaker, Dan Richman, is joining us from Johns Hopkins School of Medicine, and Eugene will introduce him properly after our center updates. This webinar is a regular event. We host the last Thursday of each month, highlighting users of the National cryoEM Centers who generously share the tips and tricks that have enabled them to make use of cryoEM in their research. You can save the date now for our talk on May 29th. Greg Pintilli will join us from Stanford University to tell us about cryoEM map model validation using Q-scores. The registration link for that is already available on the events page at cryoemcenters.org. On that site, you can also find recordings of past talks and announcements for future talks. In addition to catching up with us here online, I wanted to highlight a couple of opportunities coming up where you can meet staff from the centers. We enjoy summer meetings, so you will find staff from the centers at the ACA Structural Science Society meeting outside of Chicago in July. Including a workshop being organized by some of our staff on applications of visualization technology in the structural sciences. In addition, there's a pre-meeting congress at the M&M meeting where we hope to meet with a bunch of our users and sort of discuss what's going on with the transformative high resolution cryo-electron microscopy program. So we hope we see some of you in person there. For final logistics today, if you do have questions about making use of center resources, you can reach out to any of the panelists here using the chat. Questions for our speaker, please send those using the Q&A box during the talk. You can send them at any point, but we'll hold the questions for the end of the talk. All right, so let's get started today. First, I'll ask Vamsi to let us know what's new at PNCC.
Hi all, my name is Vamsi and I'm a microscopist at PNCC in Portland, Oregon. PNCC accepts two types of proposals, screening access and general access. Screening access proposal allows you to come on site for up to 5 days of sample prep and screening with the staff. This proposal is aimed at low resource institutions. So if you don't have a microscope, you don't have a clipping apparatus, you could come here and do your screening for the first 5 days. General access proposal awards up to 480 hours of microscope time a year for 2 years, so a total of 960 hours. This time can be used for screening or data collection. Approved general access proposals are also eligible for our training workshops, which cover topics from sample preparation all the way to data processing. Approved proposals are also assigned a scientific point of contact, or SPOC. I am a SPOC, who can assist with questions about sample optimization, data collection strategies, and so on. Our center has an a newly installed Glacius 2 and 4 cryoses. 3 out of our 4 cryoses have K3 cameras with biocontinuum HD energy filters. One of our cryos houses a Falcon 4i camera with a Selectress X energy filter. The other 3 cryoses also have Falcon 4i's but don't have an energy filter on there. For sample preparation, we have a Vitrobot, a Leica GP2, a Vitrojet, and several manual plungers, which we are actively developing and making better. So if you're interested in applying, our deadline is the first of every month. We look forward to hearing from you.
Thank you, Vamsi. And I'll turn it over to Nathan to let us know what's new at S2C2.
Good morning. At S2C2, we have our two main access programs, a high resolution data collection service. This is really geared towards labs that can prepare and optimize their own specimens, but they don't have access to a Krios level microscope to collect their high resolution data sets. Then we also have our screening and optimization program. This is geared for labs who have even less access to resources for cryo-EM, where we help users prepare and optimize their specimens. And our screening program also hosts a boot camp, which is a very intense training session for a week-long training to get really good at preparing specimens, basically developing lab hand skills. And then we also host a weekly data processing data clinic where our users can meet online and get assistance with their data processing questions. We also have a training program that hosts in-residence trainings of various lengths, as well as our workshops and lectures. We just had a nice workshop on preparing stripped avidin grids, which was really well done. We have, like most of the centers, we have our resources of our 4 Titan Krioses with various cameras and energy filters. We also have the suite of prep equipment, Chameleon to VitraJet, and of course the mainstays, the VitraBot and the Leica MGP2. We review applications monthly, and you're all welcome to come have fun with us.
Thank you, Nathan. Now I'll turn over to Ed to tell us what's new at NCCAT.
Hi, I'm Ed, coming here from New York. We offer data collection, screening, and training very similar like the other national centers. On the instrument front, we are upgrading half our instrument profile to new energy filters and direct electron cameras, for example, the Falcon 4i. So expect more to hear from that. In terms of our workflow, we are changing our workflow to incorporate coded grid boxes. So if you are an upcoming user or an active user and have not received your grid boxes, let us know. And on the training front, we are on the road quite a bit, and you heard about the summer lectures and the summer workshops. But Eugene and Christina here will be in your neck of the woods if you're in Delaware next month doing a introduction to cryo-EM sample preparation. So national centers are physical places that you can visit, but we are able to travel and go out on the road and help you in your local institution. If you want to find out more, go to nccat.nyspc.org.
Thank you, Ed. And now I'll ask Eugene to introduce today's speaker.
Thank you, Christina. It's my pleasure today to introduce Dr. Dan Richman. I know Dan as a scientist with a diverse set of scientific skills that he's very skillful with. Dan started his PhD at Johns Hopkins University, where he pioneered the use of NMR dynamics techniques with the model protein staphylococcal nuclease to study how electrostatic effects can lead to conformational changes in proteins. The insights that he uncovered are now used by the protein electrostatics field to better perform electrostatic calculations and predict alternative states of folding. Aside from being a physicist and a biologist, Dan is also a skillful communicator, having worked with the American Association for the Advancement of Science to launch a program for improving collaboration in research centers. Since 2023, Dan is now back at Johns Hopkins University as a research associate, where he has expanded his skill set to include single-particle cryo-EM, biochemistry, and enzymology, and he uses these skills now to study topoisomerases. In 2022, Dan brought his topoisomerases to work with us at NCCAT with our grid preparation and screening team, and I'm excited to hear the rest of the story today. Dan, please take us away.
All right, thank you. It's a real honor to be here and an honor to be introduced by Eugene. During a screening session, when Eugene said that I had good grid handling skills, it was one of the greatest compliments I had received in my life. So I couldn't resist an alliterative title, but a more descriptive one is that this is about the DNA binding and cleavage mechanism of DNA topoisomerase 6 or TOP6, which is evolutionarily related to a number of other interesting systems like the SPO11 meiotic recombination system and a couple of the bacterial plasmid or phage defense systems, WAJET and GABIA. So what I'll do is I want to definitely share a lot of the methodological aspects So I'm going to give a brief summary of the biological context and a quick run through of the interesting parts of our results, and then expand a little bit on some of the methods that we figured out. So the context of this enzyme is in the topological problems that arise from the fact that DNA is a double helix. So when the strands of the double helix have to be peeled apart to be read or copied, but this is taking place in a physical context where they're constrained, like a circular plasmid or even a chromosome that is in a constrained physical environment, and the ends of the DNA can't rotate. Supercoils develop ahead of, for example, the replication machinery, and then the copied double helices themselves will be entwined and linked like magician's rings, and they have to be separated to be able to be put into the dividing cells. So this is a fundamental biophysical problem. It affects all organisms on Earth, and all organisms on Earth have a set of enzymes called topoisomerases to deal with these problems. On the left side, I've named a bunch of the topoisomerases from various organisms that work on the problems that arise in replication, for example. On the right side, I show how they're classified. Mercifully, the classification scheme is pretty easy to remember. Type 1 topos cut one strand of a double helix, and they can resolve supercoils. Type 2 topoisomerase cuts two strands, both strands of the double helix, so that an entire other double helix can be passed through the break before the break is resealed. So they can resolve supercoils as well, and they can also unlink those linked chromosomes or plasmids so that they can be divided. I'll be talking about top 6. That's a type 2 topo, and specifically a subset called type 2b. Type 2as include the human top 2s that are targets of common chemotherapeutic drugs. The type 2bs are a bit structurally simpler. They lack a third gate, but they share a lot of similar architecture and mechanism. Top 6 is in archaea and plants. It has a heterotetrameric structure that's really two dimers. One dimer, the dimer of the A subunits cleaves DNA. The dimer of the B subunits traps the other DNA strand that we pass through that cleavage. That B subunit is dimerized in an ATP-dependent manner, and there's an ATP dependence to the cleavage reaction as well in ways that we don't fully understand yet. So we want to answer these basic mechanistic questions. Like we know from previous biochemical studies that it prefers to bind and act on supercoiled DNA as opposed to just any long floppy substrate, for example. How does it recognize that supercoiled substrate? Like I also just started mentioning, there's a sophisticated coordination between ATPase status and the cleavage reaction. We want to understand how that ATPase status is communicated. And like all topoisomerases, top 6 has to avoid indiscriminately cleaving the DNA. We need to make sure we reseal that break and avoid cleaving and just leaving DNA damage all over the genome. So to answer some of these questions, we wanted to visually inspect a structure of top 6 with a natural or supercoiled substrate. So the basic design of a complex here, excuse me, is to include the complete enzyme, a supercoiled DNA substrate like a plasmid, a non-hydrolyzable ATP analog like ADPMP that could lock the B subunit dimer in place, at most letting one strand passage reaction take place, but not letting the enzyme reopen and reset. and the divalent cation that is an important part of the active site. So in considering what kind of particles would work well in the thin film environment of cryoEM, we decided to use a DNA substrate that was about 10 times smaller than a typical plasmid, a DNA mini-circle instead. It's still supercoiled. As these images that don't come from our lab, but are just a really good illustration, illustrate, as you can see, like in the AFM images with the red arrows, there can be parts of a supercoiled plasmid with pronounced bends and distortions of the base pair planarity, for example. And there's a diversity in the structures of supercoiled minicircles as well. So equipped with this substrate, we solved these 5 conformations of top 6. I'm going to briefly just get to this punchline and show you some of the results, and then I'll talk a bit about the methodological trickiness of getting here. But basically, we had 2 data sets, one from NCCAT, or that was actually several data sets combined into one, and then another from our home cryoEM facility at Hopkins. And these data sets gave us a symmetric uncleaved state, a slightly asymmetric uncleaved state in which we have this slightly tilted ATPase dimer and an unfolded transducer stalk, and then a really asymmetric lopsided uncleaved state. And then the other data set was able to show us a cleaved state at a really superb high resolution that's highly symmetric, and then there a lopsided state emerged as well. You can see in all of these states, DNA is present and exhibits a severe bend of greater or lesser degree. So one of the interesting features that emerged from being able to compare all these 5 states was being able to answer one of the first questions, which was how does this recognize a supercoiled substrate? Part of the answer is that in the A subunit groove where the gate strand would bind, there are a few small loops that engage these distorted DNA minor grooves. So they're either stabilizing the pre-existing groove that is a pre-existing distortion from a supercoiled minicircle, or they are even potentially accentuating that distortion. Looking at the B subunits, where my predecessor had shown stood up somewhat unexpectedly that with biochemical techniques that there are these important DNA binding sites even really far away from the active site. We confirmed those and also found an array of several more of these clusters of DNA binding sites all along the outside of the B subunit. It was really nice to be able to compare these across these different conformations because it showed that they come into play in varying degrees as the protein and the DNA deform. So it seems like top 6 has this ample array that can be employed in different conformations depending on what the shapes of the DNA and the protein require. It's clearly very important that it stays engaged with this kind of bend, though. I'm sparing this audience any gels, but we performed supercoil relaxation assays to show that eliminating just one of these clusters, changing lysines to alanines, for example, severely damages the supercoil relaxation activity of the enzyme. So this level of DNA engagement by the B subunit is really crucial and dynamic. Another key benefit of having these states to compare against each other was we were able to see the conformational changes required to enter the cleavage competent conformation. So the B subunit is one of its domains is the transducer domains. They become an even tighter dimer and the stalks straighten, and that straightening is coupled with a slight pivot of the winged helix domain against the top rim domain. And that brings the catalytic tyrosine, which is otherwise about 10 angstroms away from the DNA, against the DNA and assembles the active site, allowing a catalytically involved metal and a few acidic coordinating residues to all come into play next to the tyrosine. So that high resolution cleavage state was really a joy to look at when we finally got it, because we could trace the backbone up to a break and then see it veer off, see that density veer off into this linkage with the tyrosine. Topoisomerases are special nucleases because they stay bound, covalently bound to the cleaved substrate. They have to be able to open the gate and then religate the substrate again. So they form this phosphotyrosine linkage, and we were able to clearly resolve it on both sides of the enzyme. So there were two interesting features that helped answer the questions of how top6 and even other type 2s potentially avoid indiscriminate DNA damage. And those are two kinds of conformational changes that are linked with the cleavage competence conformation. So actually, but before the cleavage competent conformation actually is achieved, even in just the DNA bound state, a previously unresolved long loop from the winged helix domain turns into a really ordered latch that attaches itself to the top rim domain. And that I've highlighted 3 residues there that are sort of packing residues. If those are mutated to alanines so that the packing capacity is reduced, the supercoil relaxation of the enzyme again is severely reduced. So this is a really interesting example of a coupling between a conformational change that's not close to the active site and the actual cleavage activity and strand passage competence of the enzyme. And then another important conformational change is related to that unfolded transducer stalk. So if you compare, if you look at the cleavage state and you see how tightly dimerized those transducers are and how straight the stalks are, you see that that's important for actuating the cleavage state compared to all the other states. But you notice in the what I call the tilt symmetric state, the transducers are fairly tightly dimerized, but the A subunits are nowhere near their cleavage competent state. And they're in that completely relaxed ground state. The tyrosines are very far away from the DNA, in other words. It seems like the unfolding of this transducer has decoupled a state of the ATPase that could be linked to a cleavage competent state from being able to actually actuate that state in the A subunits. This is so the stability of the transducer stalk is clearly finely tuned, because if we introduced a glycine into that stalk to destabilize the helix, we similarly saw really reduced supercoil relaxation activity. This was pretty gratifying to see on a personal basis for me, because my PhD work on using NMR relaxation techniques studied transient local structural unfolding in proteins. And it was interesting to see that as part of the mechanism of even a very large machine that we usually think of not in terms of transient unfolding events, but in terms of large mechanical motion. So it was interesting to see that come into play here as well. Okay, so moving over to the practical side of this, the really important step for getting this all to work was realizing that top6 was not going to behave on any surface other than amorphous continuous carbon. And you can see with open holes on the left, there's sporadic protein in the hole, some unfolded clusters, aggregates, it's adhering to the carbon around the edge, versus with continuous carbon that shows a broken segment, so you can kind of see the contrast. On the right, the particles are discrete, well-behaved, well-dispersed. So part of my appreciation for NCCAT was being able to narrow this down. I'd spent some time before my NCCAT sessions trying to troubleshoot all sorts of protein behavior issues within my cryo-EM grids in a pretty low throughput way. This was before the Johns Hopkins cryo-EM facility that we have now, that is in great shape and a really fantastic facility. Before it had really uh, come into, actually, I'm remembering now, I think it was during a long renovation period that I didn't have access to it. But anyway, it was up and coming. The community here too took a while to gain a critical mass of expertise. So I won't put a number on how long before 2022 I'd been working on this, but I will say it was actually a productive experience because it gave me a chance to learn hands-on cryo-EM skills with, for example, side entry cryo holders, had me figuring out how to use EPU kind of on my own, not a lot of expertise around, like I mentioned. So it was an important learning experience. Then in July and December of 2022, I did these two sample prep and screening sessions on site at NCCAT. That'll now allowed me to, at a much higher throughput, systematically test detergents, cross-linkers, grid surfaces, carbon, graphene, etc., different glow discharge or plasma cleaning techniques, even a chameleon device. And between those two sessions and some additional testing back home in the months between them, I conclusively showed that I could only use amorphous continuous carbon to get this project to work. So finally, with in December of 2022, with a large batch of grids that we made on site, I wrote a rapid access proposal at NCCAT so I could leave the grids there on site and be able to use their crioses. And those sessions were performed in the first half of January 2023. It was also interesting timing. My son was born in January of 2023. I think my grids were either going on or coming off the scope right on that day. And so I think I wrote back an email to microscopists, something like, I can't really talk about this right now, but I trust you completely. I'll also mention, you can see here in there being repeated sessions, screening sessions and repeated rapid access proposal sessions. NCCAT was really generous and flexible in allowing me to have follow-on sessions to be able to collect enough data or work out kinks in the screening, for example. So why was some of this different from the testing I'd been doing at home? The major reason that emerged that I realized was not just that it was high throughput, but that it was a different approach to being systematic in cryo-EM than I had been doing. I think a good lesson in cryo-EM is, of course, to try a lot of different things. And you don't necessarily know going into your project what will work for your system. But I think a mistaken approach that I took with that earlier on was sort of changing too many variables at once, jumping around to different parts of the parameter space without systematically changing something that could have been close to working. At the screening sessions with the coaching of Eugene and Kasahun and other staff, we really narrowed down that no, there's probably a part of the variable space that's working well. Let's fix those and then only change one thing at a time. Obviously, I mean, maybe you're laughing at me saying this is science, right? But I think when you're in a low throughput environment in cryo-EM and there's so much messiness, so much irreproducibility in cryo-EM, it's tempting to change more than one thing at a time. And what I really learned through these screening sessions was no, even though it can be frustrating and there's still a lot of messiness in cryo-EM, be systematic, change one thing at a time, and carefully move toward a grid, a particle shot that is looking good. Of course, like I said, it depends in a lot of ways on having high throughput. Being able to even do automated screening overnight, like I could do at NCCAT, for example. All right, so like I said, we collected a bunch of the rapid access proposal based CRIO sessions, they were all collected with the same setting. So I was able to merge around 50,000 micrographs overall. Around 80% of them were collected with a 30-degree tilt because in the wild type data set, we were never able to resolve or never able to fix a fairly pronounced orientation distribution problem. And so we compensated for that to some degree with just the stage tilt. Data processing also took a little bit of experimentation to figure out because especially with orientation distribution bias, there are rare views hiding, low population. They can be masked in 2D cleanup a bit, and it's very important to not throw out too many 2D classes. So we used mostly used a 3D cleanup approach called decoy classification. This is obviously a very busy slide, but I want to draw your attention to the three bolded statements on the left to guide you through this. So basically I did use some aggressive 2D cleanup to get high enough quality particles to get ab initio structures. In those ab initio references, we saw this grossly symmetric and grossly lopsided state emerge, and we're able to carry those through as references in the decoy classification. So the decoy classification using heterogeneous refinement in CryoSpark, or if you used Relion, you would use 3D classification. This was, I'm summarizing it here visually, but we used several rounds of this process, carrying forward the maps from some junk classes and the good maps, and the particles only from the good classes to keep cleaning out the junk and sorting the two useful states. So ultimately, that gave us the lopsided state and the symmetric state at higher resolution with a higher resolution consensus of the symmetric state. Then I used 3D classification in CryoSpark, which is without 3D alignments, without orientational alignments, and 3D variability analysis as corroboration to show that there was this tilted ATPase unfolded transducer substate as well. The workflow is similar for the cleavage state, but that actually popped out much easier. Fewer rounds of heterogeneous refinement, less orientation bias. So that was an interesting, subtle difference in the protein causing a dramatic difference in the quality of the cryo-EM results. Okay, so just returning to the biology for a final statement here. Like I said, these top 6 enzymes are evolutionarily related to some other interesting systems. I'm just highlighting here one of them. So one of the shared domains across some of its relatives is the toprim domain, the topoisomerase primase nuclease domain. What's interesting about what we've seen with our DNA bound structure is that we're adding to an accumulating amount of data that shows a pattern that these toprim domains bind and cleave DNA in a similar geometry, depending on this really pronounced bend. And here you can see an overlay of part of our models, but a completely different map from another system that shows where the toprim domains would fit with these severely bent DNA arms up into the rest of this Wadjet system. So yeah, I wanted to leave a bunch of time for questions. I'll just wrap up with acknowledgements here, thanking James, my boss, tremendously for resources and guidance and a huge amount of patience. Thanks to the whole Burger Lab and especially the EM and Minicircles teams. We also work closely with our collaborators, the Zukidric and Baker labs on the biophysics and structural biology of minicircles. And thanks, of course, to the cryo-EM centers that I relied on so heavily and the rest of the cryo-EM community that's been so helpful in troubleshooting. And thanks to 2 different NIH institutes for funding. All right, I'm going to stop sharing so that I can see chats and things better.
Awesome. Thank you, Dan. It's always really exciting to see so many structures. We have one question now that I can start you with while everyone else comes up with their questions. I have a few as well, but we'll start with this one. So, Valerie Lamore says, great data. Does topo 6 bind to the apex of the mini circle, or is it inducing that initial curve of the DNA?
So yeah, I think that this is I don't think that our data can resolve that question. My personal belief is that there is a pre-existing profound distortion in that kind of supercoiled substrate that this enzyme is recognizing and taking advantage of. I think I mentioned in the beginning, maybe it does accentuate that, but I don't think we can tell from this. Although maybe we could draw that conclusion from the fact that ATP binding causes, this dimerization to tighten up. Um, I will mention part of my basis for saying that I think that it is taking advantage of a severe distortion is that we actually were able to, and this is with the Zedrick and Baker Labs helps, we were able to actually identify a sequence of the mini circle that we used in our EM maps. We were really surprised because we thought it should bind indiscriminately anywhere. But when I looked at that region of the mini circles sequence, it might be a little bit more GC rich. I don't know that there's a lot of evidence out there for the bendability of different sequences, but it I think that I think that it's a stretch of the mini circle that just happens to be bent more, and then it's engaging that point.
Yeah, I have a question about the mini circles as well. Is that just like where you started with the cryo-EM, or was there earlier work where you tried other things at
Um,
like was that decision through the EM pipeline, or was that determined otherwise?
uh, a little of both. I had played around a bit with larger plasmids actually. I even saw some really nice preliminary negative stain that used them. But with the problems with reproducibility and all of the protein stability issues, I was just trying to kind of simplify it down more and more, and wound up never. When was trying that in cryo-EM, it wasn't working, and so wound up having it working with mini circles. Um, there's also my predecessor on this project had done some biochemistry with holiday junctions as well, and that's always a tempting substrate to try to get a you can with a holiday junction you have a stacked duplex on top of you have a duplex stacked on top of another duplex in a crossed geometry, which could be a way to simulate what are they called? Sorry, transport segments for topoisomerases, but those are also tricky to work with. So
Thanks. There is a question from Peter Stoyios. He said, very cool talk. Since you're working with wild type enzymes, was there a strict dependence on having non-hydrolyzable ATP? Or did you ever work with active site tyrosine mutants where you might not need the non-hydrolyzable ATP to trap DNA in the complex?
hmm. uh, I did not work with tyrosine mutants, but I did try a sample. Let me double check the wording of your question. Yeah, there there was a strict dependence on having a non-hydrolyzable ATP because if I just tried ATP, there was so much flexibility in the dimerization of the B subunits. I think because this was allowed then to cycle on and off, um, that it was blurred in cryo-EM. 2D classification wouldn't even give us um, something discrete. So yeah, I'm seeing a few questions here on the amorphous carbon versus graphene oxide grids. Should I continue with those?
Yeah, yeah, if you want to. I had the similar question. Like why did graphene or graphene oxide not work when amorphous carbon did?
I'm laughing because I feel like Eugene is probably laughing too. That like I think we were never able to answer this on any mechanistic level. I feel like probably if cryo-EM practitioners or I don't know, material scientists could answer this on a fundamental level, things would get a lot easier. Um, Let me just think if there's anything that we hypothesized. Jog my memory here. No, I can't think of any subtle chemical differences that make sense, honestly. Sorry.
I
Yeah, I
suspect the
wonder.
difference lies in like dangling bonds and imperfections in the surface.
Yeah,
Um, Something that could only probably be characterized with very careful atomic force microscopy.
right. To clarify the answer for Carter's question, the particles absolutely did behave better on amorphous carbon than graphene oxide. Yeah, and that we narrowed down in those I think especially in the time between those screening sessions when I was able to do some systematic testing again on my own at Hopkins, really conclusively just ruled out graphene oxide.
And actually, I think Vamsi has a question, but this is a related question. So if you don't mind, Vamsi, I'll
Yeah,
get
that's
to you next. But I guess, would you recommend amorphous carbon more generally for working with other topoisomerases? Uh,
Hmm.
or is that even a question you can answer?
That I don't think we even see that dependence systematically for topos, and I think other labs can verify that too. Other labs having success with like open holes with high concentrations of chapso, if I'm remembering some literature, for example. So it's incredibly subtle and different in these. I mean, I think in general you can generalize that topos are um, big, fragile enzymes, but I don't think that they all depend on having a carbon surface. Um, and uh, I but I also have a personal hypothesis that the DNA substrates that we're all trying to wrangle probably play a really strong role in this too.
Yeah, Vamsi, go ahead and ask your question.
I actually have a follow-up and a separate question also. You mentioned the DNA having an effect. I have a feeling that DNA, the charge on the DNA, is actually affecting how the particle itself is going either into the ice or staying on carbon. Um, do you have have you done any experimentations without the DNA and with DNA to see how the distribution changes, if it does?
Um, I'm trying to remember from my earliest days whether I actually did something without any DNA. I may have, and at least did really early experiments with relatively short oligos, like 70 base pair long, because like I mentioned, my predecessor on this project had, in his biochemical studies, had used those to show the importance of the B subunits being contacted by DNA, and even those did not behave in open holes.
Yeah, we recently had a proposal come through here at the NCC, and we kind of realized something similar. They had DNA in their samples, and the sample wouldn't go on to carbon; it would actually go into the holes, which was nice. But
Mm.
when they removed the DNA, the sample would just go on to carbon, which is like I hypothesize that it's probably the glow discharging and how much charge you're actually putting on the carbon. But again, there's no conclusive evidence to say this or that.
Yeah.
My second question is there a way to introduce supercoiling just by changing buffer conditions, or does the mini circle automatically supercoil, or is there what conditions does supercoil in?
Yeah, so these mini circles were expressed from a parental plasmid plasmids in bacteria are maintained at a certain supercoiling status. So if you just did a mini prep or something, those are supercoiled. these mini circles are coming out of an in vivo reaction, a transposase or integrase reaction, that when the mini circle is cut out and then religated in vivo, its supercoiling status is maintained. So they're purified in the supercoiled state. If we did various purifications, uh, like nicking and like religation reactions in the presence of different intercalators, we could manipulate the status. Some folks definitely do that a lot.
Thank you.
Right. There's a question about inhibitors, and have you worked with any inhibitors that bind the type 2 topoisomerase?
So something I didn't mention is the main presentation here actually was that my cleavage state samples were actually mixed with the toposide, but I never saw it in the structure. A toposide is well, so in topoisomerase field jargon, topoisomerase would be called a poison because it traps the cleavage complex on the DNA versus an inhibitor that is maybe working in some other part of the cycle, like blocking ATPase dimerization or something like that. So in that sense, in broad jargon terms, I use an inhibitor, but not in topo specific terms. But the etoposide, we saw like I had performed cleavage assays to make sure that I was optimizing the amount of cleavage state in the cryoEM sample. Like that was with calcium instead of magnesium, for example, and using that variant that I could achieve the cleavage state with easier. So I had some lanes in that assay, with plus or minus a toposide. And it was a very, very slight improvement in cleavage amount, but I never wound up seeing it in the structure. There's definitely a lot of interesting questions to ask about other inhibitors, though our lab is currently working on with various topos, especially ones where it's a drug that's relevant to like the human enzymes or something. That's less. Top six does have some similar sensitivities, but it's less biologically relevant. I see
All
a
right.
question. Just briefly, I see a question about using the Leica plunger. I actually
Yeah.
never used that. I did use the Vitrobot on all samples. I will make another comment on the grids. Like I said, reproducibility is really hard. Reproducibility of the thin carbon surface is really hard. I had to get very careful with using a dentin evaporator to get the right thickness. And then, as Eugene could attest to, we had to play around a lot, and Kasahun actually in that screening session too. We had to play around a lot with the strength of the glow discharge. Strong enough to get some hydrophilicity, not too strong to break a very, very thin layer.
Yeah, I was going to ask you about the glow discharge parameters and how much you had to play with those.
Yes, a lot. Because it's instrument dependent too. Like at home, I was using a Pelco Easy Glow, but at NCCAT, I think we used the Catan Solaris 2 or Solaris, and the atmospheres are slightly different. The parameterization of current or RF strength is different. It took some adjustments.
So conditions you found for glow discharging, say using the Solaris, then when you got back to your Easy Glow, you had to start over essentially, or
I
uh,
I I just had to use the principle basically that like we had hit a spot on the Solaris where it was clearly I don't know that there's a this makes me sound like an NCCAT groupie or something, but there's a parameter set there called like Laura Gold or something, and then we kept the current or the RF strength the same, and then like dramatically turned down the amount of time because it was for gold grids, which are super robust. But so it was like for many minutes or something, but we turned it down to like 7 seconds. So I used that principle over on the Easy Glow, basically kept the current in like a kind of gentle state, but then turned down the time to, I don't know, 10 seconds or something so that we didn't break the thin carbon.
all right. Uh, back away from grid type questions. There's a question about whether you would expect too much flexibility in topo in the absence of DNA. What prospects do you see for resolving a single particle structure in the absence of DNA?
Yeah, that's a great question because it would be great for us to resolve like different parts of the cycle. The prior structure, I expect a lot of flexibility without DNA, even attested to by the fact that one of my predecessors a long time ago solved a X-ray structure of a different species, top six, without DNA, and um, that the crystal structure was fine without it, but then he did some Sachs experiments with trying to see what kind of conformational changes are happening. To get it to stay in a closed conformation required a well, I just blanked on it. A disulfide, an engineered disulfide linkage. Um, so clearly they're really flexible in solution. Um, I think a way forward is, in more and more fields, people are talking about time resolved grid prep approaches. That's probably a way for this reaction cycle of topos to be parsed out more.
All right. I don't see any other questions coming in. While we make a last call for questions, I have one. So you said you collected most of your data with a small amount of tilts. How did you determine uh, what tilt to use for that collection?
I think that we tried to go as high as possible without having problematic data collection experiences. I mean, no, from doing some of this on my own. I mean, the NCCAT tilts were all collected by some of their staff, but I've done some of this on my own, and know that you can sometimes have a lot of trouble targeting um, software struggling with whole shape recognition. Also, I don't know, maybe newer versions of EPU are improving this, but the defocus isn't calibrated to compensate even for just that kind of like simple trigonometric calculation. So you wind up having to use so like if you're doing beam tilt based rapid collection, um, you have to use stage based movement instead of beam tilt based targeting, and it slows down your collection a lot. So yeah, I see questions from Valerie again. Is that why you need to tilt the grid? Yeah, as we tilt the grid because of particle orientation, the layer of carbon, I think, is definitely affecting the particle orientation. I don't have a open hole control to compare it with because the protein was just so unstable in open holes. Um, but I think that the carbon is directing it. Like I don't know if I mentioned this before. Our cleavage state was much better behaved, had less orientation bias, still was using the same carbon substrate. I think that, and it's amazing to imagine, like I described, there's this subtle conformational change in the A subunits of a sort of a more open wing helix domain orientation compared to pivoting up. It's causing a subtle difference in the charge distribution of the surface there. I guess that's enough to topple it over or something. It's a very, very subtle effect, but I think that it's having a interaction with the carbon. Yeah.
Yeah, I think if you could actually determine what minor changes were getting you a better distribution for a very similar particle and could find some general rule, you would make a lot of people very happy. But I think a
Yeah.
lot of us with experience know it's probably not so simple. Um, all right, I think we've gotten through the questions. So unless anyone on the panel here has more questions, I'd say we can wrap it up for today. All right. So thank you everyone for joining us. We hope these tips and tricks are helpful, even if they just told you you have to try all the things for your sample. But we wish everyone here luck in your cryo endeavors, and the national centers are here to support your science. So let us know how we can help you, and we hope to see you all here again.